Today I'll be teaching about genetics and health. This is thanks to a PPT given by Dr. Jazeed Mohammed Sadiq, who is a senior resident in the Department of Community Medicine, shared in SlideShare. So basically, genetics is there are two types of things. There's endogenous factors in health and diseases, and there is complex interaction between the nature and nurture. Studying genetics is effective medical and preventive intervention in many diseases. Genetic engineering helps in controlling the traits of several individuals. Chromosomes are called forms of rod-like structures. They form like X-shaped things. They are called chromosomes. Then they are present in the nucleus. So chromosome is made up of genes. Each chromosome contains genes in a linear order. They are codes for making proteins. Alternation in the genes or chromosome alter the protein produced. So there are 23 pairs of chromosome. 1 to 22 are autosomes, and 23rd is the 6th chromosome. In the somatic cells, it's always diploid because they are sperms and ova, and the germ cells are haploid. Only like haploid is basically the sperms and ova. Therefore, they'll be having only 23, whereas somatic cells, where are the normal cells, are having 46 or 23 pairs. Genes are pair of alike genes are called as homozygous, double A. If they are different, they are called heterogeneous, capital A and small a. There is a dominant gene and there is a recessive gene. Dominants are manifested in homozygous and in heterozygous state, and recessive genes are manifested only in homozygous state. Genes which affect one particular character are called polygenes. Example: muscular dystrophy, color of skin, height of and weight of a person, and the lifespan. The extent to which genetically determines condition is expressed in individuals is called penetrance. Mutation is normal genes which are converted into abnormal ones. Chromosomal abnormalities are divided into several types: non-disjunction, translocation, deletion, duplication, inversion, isochromosomes, and mosaicism. Deletion is one part of this thing gets deletion. The duplication means it will get duplicated. Inversion means it gets inversion. Substitution means one part of the gene will go into the other part of gene. Translocation means one part of the gene again goes into other part of the gene, but the other part also comes back to the initial gene. Loss of inheritance is done by a pair of genes. Unit of characteristics is provided by genes. Law of segregation says that two alleles of the same gene come from different gametes. So half of from your mother and half of from your father. Law of dominance says that when two different alleles are present, only the dominant one will be expressed. Classification of genetic disorders is divided into three types: chromosomal, Mendelian, and multifactorial, or CMM. Chromosomal or sex chromosome disorders and autosomal disorders. So there are two types of chromosomes, allosomes and autosomes. So basically there are the only two types of chromosomal diseases. Mendelian disorders can be of four types: autosomal dominant traits, autosomal recessive traits, recessive X-linked traits, and dominant X-linked X-linked traits. So yeah, all those are the different types of Mendelian diseases. They are similar to the previous chromosomal diseases in that there is autosomal and there is allosomal, and you have to remember in both of them there is dominant and there is recessive. Multifactorial diseases have more than one factors. So one of the most common one is Klinefelter, aneuploidy, abnormal males, double XY, triple XY. In nucleate males with non-functional testes, spermatozoa is absent in the ejaculate. Scanty hair growth is seen in face, axilla pubis, gynecomastia, and mental retardation is seen. XYY syndrome, male with extra Y chromosome, antisocial, aggressive, criminal behavior is seen. Height is more than 6 feet. Personality disorders are seen, in which they have behavioral disturbances. Turner syndrome, they have females with underdeveloped sex gland 45XO, non-disjunction of 6 chromosome. So they have short stature, infertile, primary amenorrhea, coarctation of aorta, pulmonary stenosis, and renal malformation. Mental retardation is also seen. Super females, which have like more than 3 X chromosomes, 3 or more than 3. Higher the number of X chromosomes, more the degree of mental retardation and congenital abnormalities. Underdeveloped external genitalia, uterus, and vagina. Autosomal disorders, Mongolism or Down syndrome is trisomy of 21, short stature, round head, tilted eyes, slits, malformed ears, short, broad hands, lax limbs, mental retardation. Congenital cardiac defects, atresia of alimentary tract. Frequency of Mongolism rise with increased in maternal age. Older the mother is when she gets pregnant, the more the chances of getting Down syndrome. Mendelian diseases can be seen in the form of they're generally seen in these types of family charts. You can see Huntington disease, which is a dominant disease. If the father is affected, then almost all the children is affected here, as you can see. Sickle cell anemia, which is recessive disorder. So a lot of them are carriers, and in this family there is only one male who is positive. Who is having pure sickle cell anemia. Hemophilia is X-linked, and only people who have only males are affected because it is also recessive, and the females are almost always carriers. So autosomal dominant traits, you have achondroplasia, Huntington's cholera, neurofibromatosis, familial polyposis coli, bradydactylia, Marfan syndrome, retinoblastoma, ABO blood group system, hyperlipidemia 1, 2, 3, 4, polycystic kidney disease, polydacrylity, then hereditary spherocytosis. Autosomal recessive disease, you have fibrocystic diseases of the pancreas, phenylketonuria, alkaptonuria, albinism, Tay-Sachs disease, agammaglobulinemia, Swiss type, hemoglobinopathies, cystic fibrosis, galactosemia, maple syrup urine disease, and Hutch-Springs disease. Recessive sex-linked diseases, you have hemophilia A and B, Duchenne muscular dystrophy, color blindness, G6PD deficiency, hydrocephalus, retinitis pigmentosa, agammaglobulinemia, Bruton type. Dominant X-linked traits, vitamin D resistant rickets, and familial hypophosphatemia, blood group 10XG. So blood group of O is seen in diseases like duodenal ulcer, gastric ulcer, hemolytic tendencies. Blood group of A have more chances of getting stomach cancer, uterus and cervical cancer, pernicious anemia, thrombosis. Next we have erythroblastosis fetalis, Rh positive fetus in Rh negative mother. First time is not a problem because first time the mother's blood is not sensitized to Rh positive blood. Or the second delivery onwards, it will be very much sensitive. There are two types: Rh antibody, strong or saline antibody, weak or albumin antibodies, which cross the placental barrier and passes back into fetal circulation. Fetal RBCs are destroyed, death in utero, or they are born with jaundice, anemia, and edema. Okay, then you have sickle cell anemia, which is recessive disorder, abnormal hemoglobin, point mutation in DNA. Heterozygous individuals are clinically healthy. Homozygous have acute hemolytic anemia. Rate of sickening depends on hemoglobin S present in RBC. Disorder is from first year of life when HbF falls. Delayed puberty, chronically ill, jaundice, hepatomegaly, enlarged heart. There is no specific treatment. Prenatal diagnosis and genetic counsel for couples that are at risk is required. Thalassemia is reduced synthesis of globulin chain alpha or beta, or reduced hemoglobin synthesis. Hypochromic microcytic anemia. Alpha thalassemia is associated with gene deletion, whereas beta thalassemia is due to point mutations. Signs develop after 6 months when hemoglobin synthesis shift from hemoglobin fetus to hemoglobin adult. Prenatal diagnosis is required for the couples who are under risk of all this. Again, it is having hypochromic microcytic anemia, decrease in color, decrease in size of RBCs. Okay, then we have hemophilia, which acts again, which is X-linked, recessive disorder. It affects only male, and the female are always carrier. Deficiency of blood coagulation factor is seen, and there is prolonged bleeding in large joints. Progressive disease, joint disease, and muscle atrophy is seen. Cystic fibrosis, gene defect affecting respiratory and GI tract and sweat glands. The treatment in cystic fibrosis is gene therapy. Phenylketonuria, there is deficiency of liver enzyme phenylalanine hydrolase. Therefore, phenylpyruvic acid is excreted in the urine. Phenylalanine accumulates in the blood and tissues, which is having a toxic effect on the brain, causes mental retardation. Elevated levels of phenylalanine is usually indicative of this. Bottle fed in infants tested after first successful formula, which is more than 48 hours. So breastfed infants should be tested after 7 days. Multifactorial diseases are having polygenic inheritance. They are controlled by a number of genes located in different sites. Mode of inheritance is complex because the environmental factor is also involved. Examples are diabetes, hypertension, ischemic heart disease. Genetic predisposition is also more common for adult in life. Like in cases of cancer and coronary heart diseases, diabetes, hypertension, and mental disorders, premature onset of common disease is seen. Family-oriented approach with appropriate therapies is also useful. Advances in molecular genetics, DNA technology, gene therapy, human genome project, human genome diversity project. Gene therapy. Basically, this is involving gene sequence, like the process of putting a gene sequence into a cell with the aim of modifying the cell's behavior to in a clinically relevant fashion. Gene introduced is via virus, which is retrovirus or adenovirus, or lipid or receptor targeting. Inserting a patient into gene into patient cell can become alongside other treatments in the future. Okay, so there are like several therapy. Gene mutation therapy is there. Gene inhibition therapy is there, where the new gene inhibits the mutant gene. Or there is gene suicide therapy, where the cell itself will get destroyed in the cases if it is formed. The mutant cell will get destroyed. Okay, so approaches to gene therapy, you have in vivo gene therapy. Normal genes are delivered inside the body. And you have ex vivo gene therapy. Genes are inserted inside the cells outside the body. So first it is removed from the body, then the gene is inserted, then it is put back into the body. So this treatment is usually done for cystic fibrosis, hemophilia, muscular dystrophy, sickle cell anemia. Bacteria containing plasmid with cloned normal human gene and genetically disabled retrovirus cloned is this both of them will combine and will become incorporated into the virus. T cell with disabled ADA gene isolated from SCID patient. They are taken from, this is ex vivo gene therapy, so they are taken from the patient. The retrovirus transfers the ADA gene to cell. The new cells with the new genes, the new WBC with the gene are taken, and they are cultured to ensure that the ADA gene is inadequate. The genetically altered cells are re-implanted to produce ADA. This is for SCID, severe combined immunodeficiencies. Okay, the next you have Human Genome Project. This is systemize the research on mapping and isolating human genes in order to create a single linear map of human genome with each gene coding gene defined and sequenced. Aim is to determine the complete sequence of nucleotide base pairs that make up human DNA and the gene that it contains. So this is done by the coordinating Human Genome Project data, which is done by UNESCO, Genome Database, HUGO, National Institute of Health or Department of Energy of USA, Medical Research Council of the UK, GeneThon of France, and European Union. This was started in 1990, and it is also called as a 13-year project because that's how long it took. By April of 2023, it is 92.1% completed. So how a easy way to remember HGP in coordinating the data are UGHNIMGE. Basically, UNESCO and Genome Database, HUGO, Medical Research Council, and they're the main important parts. So UNESCO you remember, HUGO you remember, Genome Database you remember, and Medical Research Council of UK. Hardy-Wahlberg equilibrium. This is a theory in which allele and genotype frequency in a population will remain constant from generation to generation in absence of other evolutionary influence. Population genetics states that it is basically the study of precise genetic composition of a population and various factors determining the incidence of inherited traits in them. So the factors affecting human gene pool are mutation, natural selection, population movements, and breeding structures. Mutation means UV radiation, the gene mutation will be there. Natural selection is Darwinism. Some of them will die, some of them will live. The people who live will pass their genes. Population movements is from one place to another, and breeding structure is who the people reproduce with. So what about preventive social medicine? Okay, so primary prevention is health promotion, specific protection. Secondary prevention is early diagnosis and treatment. Tertiary prevention is disability limitation and rehabilitation. Health promotion, this is not really possible, but you should you should be aware of it. There is euthenics, genetic counseling, and other genetic preventive measures. Eugenics is basically the aims to improve genetic endowment of population. Controlled breeding increases the occurrence of desirable characters. Positive is for desirable traits like fit people to have children, in vitro fertilization, cloning. Negative eugenics is reducing the undesirable land. Example, Hitler killing the weak and defective to improve German race, and sterilization to reduce frequency of hereditary diseases and disability. Euthenic is the study of improvement of human functioning and well-being by improvement of living condition. This is environmental manipulation. Educational and controllable environment prevention and removal of contagious parasites, employment, housing, and sanitation. There is genetic counseling, like individuals and families should be counseled to understand the medical, psychological, social, and reproductive implication of genetic and congenital condition. They have to determine the facts, transmit the information, and support the decision of the parents. There is prospective genetic counseling and retrospective genetic counseling. Genetic defect has not happened yet. True prevention of disease is prospective genetic counseling. You have to identify heterogeneous individual for any particular defect, and you have to prevent heterozygous marriage. One of the two common diseases are sickle cell anemia and thalassemia. If both the parents are having, then they should be counseled to not marry. There is also retrospective genetic counseling. These are hereditary diseases which are already occurred in the family. This is most common. Contraception, pregnancy termination, and sterilization may be required. Most commonly, congenital abnormalities, mental retardation, psychiatric illnesses, inborn errors of metabolism is seen. Other genetic preventive measures are prevent consanguineous marriages, avoid late marriages, and avoid pregnancy among women more than 30 years of age. So specific protection. So how can protection of individuals and communities from mutagens, X-rays, ionizing radiation, chemical mutagens, anti-D globulin, Rh hemolytic disease. Then early diagnosis and treatment, detection of genetic carriers, elevated creatinase in muscular Duchenne dystrophy, prenatal diagnosis, screening of newborn infants for CDH, phenylketonuria, Duchenne muscular dystrophy, congenital hypothyroidism, and CF, recognizing preclinical cases in cases of type 1 diabetes, phenylketonuria, and thalassemia. Okay, so if a patient is having elevated creatine kinase, then you can say that they are possibly genetic carriers of Duchenne muscle dystrophy. And then you have prenatal diagnosis. And this is indicated in advanced maternal age, previous child with chromosomal problems, intrauterine growth delay, biochemical disorders, congenital anomaly, screening from neural tube defects, and trisomy. Methods are cryogenetics, amniocentesis, chorionic villi sampling. Amniocentesis is done in 14 to 16 weeks. Chorionic villi sampling is done in 10 to 13 weeks. Protein assay can be done. DNA diagnosis can be done. Sonography, fetoscopy, maternal serum alpha fetoprotein, chorionic gonadotropin. So disability limitation, which is a tertiary form of treatment. In case of phenylketonuria, low phenylalanine diet. In case of hemophilia, sickle cell anemia, thalassemia, therapeutic and preventive measures must be taken. In cases of rehabilitation, in congenital heart defects, cleft lip and palate, neural tube defects, surgical treatments must be done. Teaching skills through aids in cases of people who are having mental retardation. There is another thing, concept called as Ashwa Sari Saki Iranam, in which the assistance is given to the caregivers. Genetic properly, again, these are the screening and this thing which can be done. Primary prevention in cases of RH diseases, congenital rubella, congenital malformation. Preventive or screening action, postpartum anti-D globulin, immunization of girls, maternal folic acid, control of maternal diabetes mellitus, avoidance of mutagens and teratogens. Antenatal screening, congenital malformations, chromosomal abnormalities, inherited disease. Neonatal disease, you can perform fetal anomaly scan, alpha fetoprotein estimation, maternal age, maternal serum factor levels, family history, screening for hemoglobinopathy, Tay-Sachs disease. Neonatal screening, you have congenital malformation, phenylketonuria, congenital hypothyroidism, sickle cell disease. You can examine the newborn for early treatment. Biochemical tests for early treatments. Thank you. Please check out Jazila Mohammad Sadiq. Very huge thank you for providing their PPT online.